Okay, we are live. Mute kan? Unmute kan?
Unmute jam doa.
Dah mute ke unmute?
Unmute.
Apa Datuk? Assalamualaikum.
Nak unmute.
Right, good.
Okay. Sorry to interrupt. We are live. Can
Yeah,
you guys
okay.
please mute your mic?
Dah boleh cakap dah.
Okay. Tutup. Emma. Mute.
Betul.
Live dah.
10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0.
And mute your speaker during all session. The conference local chair, our invited speakers, distinguished guests, ladies and gentlemen. First of all, it is my pleasure to be the MC for the Asian Symposium on Medicinal Plants and Spices, ASOMPS 17, with a tagline of contextualizing natural product science for societal relevance. My name is Fatima Salim from University Technology Mara, representing the Malaysian Natural Product Society, MNPS. On behalf of the organizer, we wish to extend our warm welcome, Selamat Datang, and thank you to all participants, guests of honor, delegates, representatives, as well as the foreign and local participants to the 17th ASOMPS, and this is first time as visual conference 2021 hosted by Malaysia and organized by the Malaysian Natural Product Society. Ladies and gentlemen, to seek blessing for our event today, I would like to invite Ustad Muhammad Safwan to lead the dua recitation. Please welcome.
Thank you, MC, for the introduction. Al-Fatihah. بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على أشرف المرسلين وعلى آله وصحبه أجمعين. Ya Allah, Ya Rahim, praise be to God, the Lord of the world. Peace and blessing be upon our Prophet Muhammad sallallahu alaihi wasallam and his companions, and for those who follow his example till the judgment day. Allahumma Ya Allah, on this blessed morning, in conjunction with the 17th Asian Symposium on Medicinal Plants and Spices, we beseech, pray, and grateful towards you in favor of all the infinite blessing to us, your humble servant, to live in safe and prosperous life. We seek your blessing for a flawless progress of this event from the beginning till the end. We seek your guidance to steer clear of events that would detrimental the progress of this event. Ya Latif, Ya Rahman, please bless us with your tawfiq and hidayat. Please guide all of us to greatness, peace, glory, and prosperity in this world and the hereafter. Make us a responsible intellectual. Grant us with a valuable knowledge that will be beneficial to mankind in order to gain your mardatillah. Make us your righteous servant that followed your commands and neglect the sinful act. Please forgive us for our wrongdoing. Ya Munzil al-Barakat, bestow peace to our beloved country, Malaysia. Preserve us from any threats and disaster, neither man-made nor natural disaster. And to you, Ya Allah, we ask for security and prosperity upon us, our leaders and our country. Bestow patience in us in order to face the challenge from you. Please accept our deeds and please reward us accordingly. ربنا عليك توكلنا وإليك أنبنا وإليك المصير. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار. وصلى الله على سيدنا محمد وعلى آله وصحبه وسلم والحمد لله رب العالمين.
Amin, amin, ya Rabbal Alamin. Thank you, Ustaz Muhammad Safwan. Distinguished guests, ladies and gentlemen, we are pleased to inform you that ASOMPS 17th conference will feature 4 plenaries, 16 invited speakers, 64 orals, and 52 poster presenters, with more than 40 participants that include local and international researchers, industrial experts, as well as professionals. To begin, I would like to invite Professor Dr. Nur Hadiani Ismail, the President of the Malaysian Natural Product Society, to deliver her welcoming speech. Please welcome.
Assalamualaikum warahmatullahi wabarakatuh. Good morning. On behalf of the Malaysian Natural Product Society, I am really pleased and honored to welcome all distinguished guests and participants to the 17th Asian Symposium on Medicinal Plants and Spices, ASOMPS 17 in Malaysia. It is really indeed a great honor to be hosting this meeting. And for the first time, we are organizing this collaboratively involving several universities and research institutes in Malaysia. Since its inception in Peshawar, Pakistan in 1960, ASOMPS has been organized biannually in Asia to promote networking in natural products research. Since then, much had been accomplished in fostering regional research collaboration and cooperation among the Asian scientists in chemistry, pharmacology, pharmacy, biochemistry, botany, and biotechnology of medicinal plants and spices. This meeting was supposed to be held last year, but due to the global COVID-19 pandemic, it was postponed. And for the first time, our meeting is now conducted virtually. It has indeed been a steep learning curve for us to learn how to organize an online meeting. The theme for this year's meeting is contextualizing natural product science for societal relevance. It aspires to promote a paradigm shift as to how natural product science throughout Asia is pursued and conducted to efficiently harness endemic resources towards the sustainable translation of scientific findings for human benefit. In particular, Malaysia is blessed with a plethora of biodiversity. It is estimated that at least 15,000 species of vascular plants in Malaysia, with at least 3,000 of these plants having medicinal values. There are countless research in natural products in the region, and yet very few reaches the patient's bedside. This meeting is hoped to promote translational research, converting sustainable research findings from the plants to the bedsides. They also serve as a platform to foster stronger research collaborations. In drawing up the scientific program for this meeting, we are indebted to Professor Jeffrey Cordell for his constructive advices through numerous discussions with the committee. We are grateful to all our distinguished plenary and invited speakers who have graciously invited our invitations to speak at this symposium. And finally, I would like to acknowledge and the much appreciated support from Institute Kimia Malaysia, Phytochemical Society of Asia, JAI, Japan Analytical Instruments, and all participating universities, UPM, UITM, UKM, and many more in Malaysia, and the research institutes for making this symposium a success. I wish all participants an enjoyable and fruitful meeting. Wassalam.
Thank you very much, Professor Dr. Nur Hadiani Ismail, for the warm welcoming speech. Next, it is my pleasure to invite Professor Emeritus Dr. Nordin Haji Lajis, the Honorable Member and one of the co-founder of the Malaysian Natural Product Society, to deliver the opening speech and to officiate the ceremony. Let us watch the video that Professor Emeritus Dr. Nordin Haji Lajis had prepared.
Bismillahirrahmanirrahim. Hello everyone. Assalamu alaikum. May peace be upon you. Welcome to this important event. It is gratifying to see the lineup of prominent speakers who I consider to be the true custodian of natural products, as well as the young and new faces in this conference. By this representation, I believe this is going to be a great and useful occasion. I wish to thank the organizer for giving me the opportunity to welcome the participant, and at the same time, I also like to congratulate them for their work that make this conference a reality. ASOMPS has been an important event closely associated with the history and progress of natural product research and education in Malaysia. ASOMPS 8, held in Malacca in 1994, marks the beginning of more organized effort in promoting natural product research in Malaysia through the formation of the Malaysian Natural Product Society in 1995. The society, which is the organizer of this conference, has been continuously active in pursuing its mission since its formation. ASOMPS 8 was in fact the first major international conference on highly scientific topic ever held in Malaysia at that time. Here, I would like to record the support of the late Professor Jack Cannon of the University of Western Australia in promoting natural product research in this country during its early days. His contribution has led to the success of ASOMPS 8 and eventually the formation of the Malaysian Natural Product Society. The salient issue during ASOMPS 8 was on the protection of biodiversity and the sharing of benefits from biodiversity. The topics was deliberated during the conference. Although we may have lost the struggle due to economic and commercial forces, the effect of deforestation and loss of biodiversity has become more obvious now by the current pandemic and climate change. Despite the main concern then was the loss of potential new drugs or drug models, the indirect consequence of deforestation and loss of biodiversity cannot be dismissed. Malaysian Natural Product Society was established with its major purposes, among which include to promote research in natural products and to elevate it to international standing, to promote forums, dialogues, and interactions between fellow chemists, interaction with researchers from different disciplines, as well as with other relevant agencies. And thirdly, to promote and enhance collaborative integrated research. You will see that the term natural products was purposely adopted in the name of the society to allow membership from different disciplines while implying that chemistry as the fundamental element of science. Natural products has come a long way since ASOMPS 8, mainly driven by the accessibility and improvement of numerous hard and soft tools as well as gadgets. We are grateful for being able to tag along with the progress in the science and techniques in natural products, mainly through assistance from as well as collaboration with our brothers and sisters from better established institutions of different countries. New ways of doing science have emerged and continue to grow, generating new concepts in science or verifying old ones not proven before. Natural products, being a fundamental and established field of study, can contribute to further progress of science and technology. However, for this to happen, one has to work with others. We contribute our expertise to others while we adopt the knowledge of others in our work. The future is wide open for everyone to explore through persistence, collaboration, and cooperation. I hope the interaction among participants in this conference will generate new inspirations, especially to the young and upcoming scientists, and encourage cooperation. I hope that you will enjoy the exciting presentation by the distinguished speakers to come. Finally, I pray for the success of this conference and hope friendship will be strengthened henceforth. Thank you.
Thank you very much, Professor Emeritus Dr. Nordin Haji Lajis, for the meaningful speech and officiating ASOMPS 17, 2021. Next, I'm honored to invite Professor Dr. Barry Noller, the head of International Advisory Committee of ASOMPS, for his welcoming address. Please welcome Professor Dr. Barry Noller.
It is my very great pleasure to extend to the guest of honour, all distinguished lecturers and delegates, a very warm welcome to ASOMPS 17 here at Putrajaya, Malaysia, following ASOMPS 16 held 6 to 8 December 2018 at Colombo, Sri Lanka. This is an appropriate venue for the ASOMPS conference in Asia on medicinal plants and spices and natural products, as Malaysia is one of the biodiversity hotspots in Southeast Asia and linked to natural products delivering therapeutic benefits. ASOMPS 17 now follows ASOMPS 8, held in 1994 at Malacca, whose chair was Professor Nordin Lajis, who tabled the Malacca Accord and subsequent Kuala Lumpur guidelines that followed the Manila Declaration, the profoundly effective outcome of UNESCO with ASOMPS 7 at Manila in 1992. ASOMPS carries its banner of promoting research on plant resources, phytochemistry, and pharmaceuticals and natural products chemistry since its humble beginning under the guidance of Professor Finn Sandberg at Peshawar, Pakistan in 1960. It is clear that ASOMPS as a regional symposium has met its objectives to promote a spirit of collaboration and cooperation for the benefit of scientists in Asia, to be a window for the rest of the world, demonstrating what scientists in Asia are doing and what they may be able to do, to take pride in the achievements of scientists in the Asian region, to provide a venue for young researchers to meet with peers, international experts, and more senior colleagues, then enabling a forum for career development. These objectives remain at the core of the ASOMPS concept, functioning as guidance for those who organise the meetings and indicating to the international scientific community and its sponsors the general goals of the meetings. 16 meetings of ASOMPS have now been held. Clearly, ASOMPS has an important and continuing role in the future. Now is the 17th symposium and the second time in Malaysia. The ASOMPS 17 symposium focuses on contextualizing natural product science for societal relevance, covering pharmacology, medicine, clinical evidence, biochemistry and chemistry, sustainability of bioresources, industry applications, and bioinformatics, data mining, and artificial intelligence. Professor Intan Safana Ismail and co-workers have put ASOMPS 17 together with a lot of effort, including the complications introduced by the COVID-19 pandemic. I now wish all invited speakers and delegates, both local and foreign, a very fruitful and interactive conference. Thank you.
Thank you very much, Professor Dr. Barry Noller, for your welcoming address. Alhamdulillah, we are now ready to begin our very first plenary session. It is my pleasure to invite the Honorable Member of Malaysian Natural Product Society, Emeritus Professor Datuk Dr. Lailidin, to introduce our speaker. Please welcome Datuk Dr. Lailidin.
Thank you, Dr. Fatima. Assalamualaikum and a very good morning to all participants of ASOMPS 2021. A very good morning to Professor Norman Lewis. I think now it is around 6 o'clock in the evening in Washington. I am Laili, and I have the honor to be the moderator for the first plenary lecture of this symposium. Our first plenary lecture will be delivered by Professor Dr. Norman Lewis from Washington State University. Professor Norman Lewis is a Washington State University's Regents Professor and a Fellow of the Institute of Biological Chemistry, a Fellow of the Royal Society of Edinburgh, the Arthur M. and Kate Isaac Todd Distinguished Professor, and Professor Norman Lewis obtained his PhD from the University of British Columbia in 1977. Professor Norman Lewis will deliver the first plenary lecture of this symposium, and the title of his lecture is The Region Protein Roadmap to Diverse Medicinal Health Protecting and Plants Defense Plants Phenol. Now I would like to invite Professor Norman Lewis to deliver his lecture.
Thank you very much. Good Good morning to everybody. It's just beginning early evening here, and hopefully you can see the slides as we begin. So thank you for the very kind introduction. As you mentioned, the title slide, but what I'm really trying to emphasize today is that detergent proteins are the sort of gateway entry points, if you will, to a variety of different plant phenols of having medicinal, health protecting, and plant defense functions. So, as all of you know, that the evolution of plants began with sort of green algae, which is shown here in the lower left-hand side, and then as the algae then began to get onto land, we began to go through charophytes, through other organisms such as liverworts, then mosses and hornworts. Of course, Professor Asakawa, who's speaking later, had massive contributions in this particular area. And then evolution of plants went to club mosses, ferns, gymnosperms, and flowering plants. There's now about 390,000 different species. And the real key aspect from our perspective anyway, in terms of land plant colonization, was the evolution of plant phenol biochemical pathways, particularly phenylpropyroid metabolism. And just to sort of give you an indication of what all of that is, the aspects are, if it weren't for the plant phenols, certainly the vascular plants that we have in existence today would not be able to be present. I'll indicate the reasons for that slightly as we move along. In some plant tissues, the amounts of plant phenols can be massive. It can be 50% or more. And as I'm going to point out, these detergent proteins are gateway entry points to distinct, many of them are bioactive plant phenol classes. One aspect that you can think of in terms of land plant colonization is the ability to be able to have the systems able to live and thrive in a desiccating habitat, and that occurred through biochemical processes that evolved, including in large part the phenylpropyroid pathway, suberin and suberization. And of course, these have given results, not only help to protect plants, thrive and survive in drought environments, but the products of suberin cork, for example, cork rings and stoppers, etc., are important natural products that we use on a daily basis. Another plant phenol that you can also consider as being responsible for land plant colonization is lignification. This is probably second most abundant natural product on land plants, 20 to 30%, depending on the types of species. And it is responsible for plants being able to stand upright, grow to extreme heights, and they can, the lignified vessels and conducting systems enable water and nutrient attack. But from a bioactivity perspective, the generation of lignification has provided a physical barrier to a variety of microbes and herbivores, foraging, etc. So these are the two of the important land plants that were certainly of plant phenol classes that are certainly responsible for the evolution of land plants, in the 390,000 or so that are there. I'm going to focus, though, instead on what I'm calling bioactive vascular plant classes. These are moieties like lignans, various diterpenoids, pterocarpins, stilbenes, and I'll finish up by focusing a little bit on elygiotannins and proanthocyanidins, sometimes known as hydrolyzable and condensed tannins. So when you consider the aspect here of vascular plant evolution, the lignans and the lignans and the ability to get onto the terrestrial environment is very much, a key feature of this aspect. And as you go from the lycopods through to the angiosperms, you can get these three hydroxycinnamyl alcohols, paracumaril alcohol that doesn't lack methoxyl groups, coniferol with 1, and synapyl alcohol with 2. And these processes evolve between the lycopods coming into play until the angiosperms. But in terms of the lignans and the neolignans and lignans, I think it's worthwhile making a few points here about some of the pioneers that worked in this area. Richard Haworth, who passed away in 1990, came up with the term lignans to talk about phenylpropanoids that are linked through 8,8' bonds. And these are shown in the two structures below here with 8,8 linkages in both instances. Holger Ertmann, though, who was a 1973 chairman of the Nobel Chemistry Committee, came up with the concept of radical coupling or dehydrogenated polymerization, giving rise to molecules like lignans and lignans. And he is certainly responsible for that key discovery. Ted Hillis, who I show in this picture here from Australia, worked on trying to understand the compartmentation of a lot of these moieties. Some were lignans, some were flavonoids, etc., and showed that some of these low molecular weight bioactive compounds could accumulate nearly in crystalline form and sometimes nearly pure forms in different cells and cell types that are present in vascular woody plants. And the other person here, Otto Gottlieb, who unfortunately also passed away in 2011, came up with the terms of neolignans to more or less cover every other kind of coupling except for the 8,8'. And just a few examples here, 8,5, 8,1, 7,1'. So ourselves here, our interests here are not just in lignan and lignans and say pterocarpans, but also allophenol biosynthesis. So the allophenols and propanol phenols that you can see here that are characterized by molecules like eugenol and isoeugenol, these are important phenylpropanoid constituents that are present in a variety of moieties that are used here in the United States, for example, in mouthwashes and a variety of different aspects. And we looked at the biochemistry of these systems to try and establish what these biochemical pathways actually were. And here you can see from the allophenol and propanol phenol, sorry, excuse me, this is moving its own. You can see here that the first step in generating allophenols and propanol phenols is the activation here of the hydroxy, the alcohol group here to form a good leaving group here, and then these allophenol synthases and propanol phenol synthases that we reported and patented some years back, then enable you to be able to generate either the chavicol, the allopropanil phenols, or eugenol, or the corresponding isomers, para-enol and isoeugenol. So that's given us a entry point into being able to biotechnologically modulate their levels in different plant species. But I'm going to focus here on land plant phenol classes and dirigent proteins. We introduced this term almost 20-something years ago, dirigent from the Latin dirigere, and its meaning is to guide or to align. And I'll try to explain how that comes into play as we move along here. So in terms of the chronology, since we first detected stereoselective coupling, and I'll go into this to form a molecule called +pineresinol, we then were able to purify the corresponding protein. We called it a +pineresinol forming dirigent protein at that time. We cloned the corresponding gene in 1999 and then showed that this was on the pathway to podophyllatoxin, which is a scaffold for one of the most important plant anti-cancer compounds. And we did a variety of other aspects looking at in Thuja placata, where its gene was being expressed, looked at some of the biochemistry, and then we introduced the concept, a hypothesis in 2006 that dirigent proteins could also be involved in producing the lignin polymer. Well, then later by 2008, aromatic diterpenoids such as gossypol were shown by our collaborators, Bob and Dr. Liu, Bob Stepanovic. And then by 2010, we found another divergent protein that could form the opposite antipode of pinoresonol with minus form. By 2013, the entry point with divergent proteins into pterocarpans began to emerge, as did evidence for lignification and divergent protein formation, high molecular weight oligometic lignans, divergent proteins, and these crystal structures of these systems have been described by ourselves in 2015 and more recently in the last couple of years. And just this year, the very first neolignan, which is an 804 linked lignan, was reported as well. So these are all different gateway entry points into different plant phenol classes, as I'm going to focus upon. So the question here, when we looked at the family of divergent proteins in most plant species, we could see there are about, from A to H, different subfamilies, and these families can generate lignans such as pinoresonol, aromatic diterpenoids, which I've mentioned, pterocarpans, and also down here in DIR-E, the lignin biopolymers. The question here is how stereoselective divergent protein mediated control of radical coupling occurs. In the presence of an oxidase, which is considered generally a peroxidase or a lacase catalyzed reaction, you can form free radicals. In the absence of the divergent protein, you get different forms of coupling with different racemic products that are generated. But in the presence of the divergent protein, in this case, a plus pinoresonol forming divergent protein, we get only psi-psi coupling of these two free radical forms in quinone methide form, which then cyclize to give rise to plus pinoresonol. So when we looked at doing work with the divergent proteins, we require a complementary oxidase, or an auxiliary oxidase, generally lacase or peroxidase. And you can see here that as we increase the concentration of one DP that forms a plus pinoresonol, this is from Schisandra chinensis, or these ones here from Arabidopsis, DIR6 and DIR5, you can either get increasing in antimetic excess of plus pinoresonol or of the corresponding minus antipode. So that was the first system of showing that a divergent protein was controlling the outcome of phenoxyradical coupling. So we can see that we've got two different types of stereoselective coupling. We've got the plus pinoresonol forming protein, which generates, in the one case here, the psi-psi coupling to give the plus antipode, or the ray-ray coupling to give rise to the corresponding minus form. Both cases, you're forming a bis-quinone methide intermediate, which then undergoes carbon-oxygen formation to a carbon-oxygen bond formation to form either the plus or the minus pinoresonol forms. So that we compared the gene sequences of the plus pinoresonol forming divergent protein, which we call SCDRR here from Schisandra chinensis, and also the one from that makes minus pinoresonol from Arabidopsis, here DIR6. And we identified two domains, domain A and domain B, and we swapped these systems out, but only in the case of domain B, where we're able to see each switch in stereoselectivity. So here you can see the assay that we are using, the plus pinoresonol forming divergent protein that's shown here. The assay here generates, under the conditions formed, in excess of the plus antipode over the corresponding minus. The Arabidopsis one, by comparison, forms the corresponding minus form rather than the plus form. But when we take the divergent protein from Schisandra chinensis, that domain, then we switch what we saw before with the native Schisandra chinensis to now having the system now going from being plus pinoresonol forming system to a -1 by switching out that region in the AT-DIR6. So that gave us some indication as to where the important aspects were within the active site that's responsible for the different forms of stereoselective coupling. We completed the three-dimensional structure of the first divergent protein to be crystallized at 1.9 angstroms. And you can see here from the slide here, this is a trimeric form that's shown here, top side and full view that's shown here. It's an interesting protein in the sense of the entrance to the active site is shown here. It's got some phenylalanine moieties here that are that we believe are involved in other protein-protein interactions, which I'll come back to later on. And within the active site, this shows the form of coniferyl alcohol, either as coniferyl alcohol as a free radical form, and the corresponding amino acid residues that we believe are responsible for binding the quinomethide radical intermediate and then enabling the side coupling to give rise to the corresponding plus antipode. Well, what other aspects of pinoresinol might be of interest is that we've looked at the biochemical pathway beyond pinoresinol in plants like Podophyllum peltatum, Podophyllum hexandrum, which produce this molecule, podophyllatoxin. We showed there's a sequential reduction with an enzyme here called pinoresinol reductase that forms first larisresinol, sequoisolarisuresinol. This can be dehydrogenated with the corresponding dehydrogenase, and then we can generate the methylene dioxide bridge here with a cytochrome P450. And then there's a variety of other steps that are quite well known now that get us into podophyllatoxin. And as I mentioned earlier, it's a scaffold for the semi-synthesis of etophos, etoposide, teniposide, etc., that are widely used in the treatment, successfully widely used in the treatment of various cancers. So I thought I might just show a few slides of looking at trying to understand in Podophyllum hexandrum and the rhizome, kind of root tissues, as to where podophyllatoxin accumulates. So we use a MALDI imaging here, and you can see that in the vascular bundles, which are shown here in the dark, there's very low abundance of the podophyllatoxin in the presence of potassium ion, whereas it's in high ion abundance in the pith in this region here and also in the neighboring parts of the cortex. Conversely, though, when we look at camphorol, a flavonoid here, you can see it accumulates exclusively, almost exclusively here in the vascular bundles, but it's absent in the pith and present to a very low extent in the corresponding cortex tissues. So that just shows high abundance in the vascular bundles, low abundance, noise level in the pith. And we can distinguish between isobaric molecules like potassium and podophyllatoxin.
Yeah, back.
We can distinguish between isobaric moieties like podophyllatoxin and beta-poltatin by using ion mobility processes here, and we can look at the localization and the cross sections here again of the rhizome, showing the rhizome with the pith, vascular bundles, etc. And you can see where the beta-poltatin accumulates relative to the corresponding podophyllatoxin. So that brought our next attention to, well, where are the genes being expressed that are generating molecules like podophyllatoxin? So before I do that, though, this is just an overlay slide here showing the podophyllatoxin corresponding glucoside and the camphorol shown in these different colors. You can get these wonderful localization pictures showing where these molecules exist in these different particular plant tissues. And this is quite, for me, rewarding, given that when I first began working in natural products, we just used to take dry material and grind it up and had no idea where anything came from, but was always delighted when we were isolating some molecules. But now you really get to see what's going on at a different level. So we use laser microscope microdissection. I'll just try and get this to go to show what I'm meaning here. We use this system here to cut out different cell types. This is an example here, just cutting through Actually, in this case, it's an Arabidopsis. It's not the podophyllum that we're looking at, but we're cutting out here the vascular bundle here, and then we pop it out, as you can see, and then collect that. You do that several hundred or thousand times, and you've got enough materials for chemical analysis or to look for RNA samples, as I'm going to show next in this next slide. So here we use laser capture microdissection. We cut out cortex, xylem, and pith samples as shown here. We extracted the RNA, the cDNA synthesis to see where the genes that were involved, in this case, where the genes were involved in generating these lignans that are shown here. And this is shown in this slide here. You can see these are the vascular bundles that are shown here. This is the sort of cortex region. This is the pith. I showed you before where the podophyllatoxin is, but you can see that the label, or should I say the gene expression, is in the vascular bundles. It's not actually where it's in these regions here. It's not actually where the podophyllatoxin itself is being generated. So the precursors to podophyllatoxin have been formed in the vascular bundles, and then the final stages of the biosynthesis and accumulation, the molecules are being transported into the pith and into the cortex. So then I'm going to switch away from that form of bioactive compound to turn our attention to flax, which is an interesting, this was a paper we published just a few years back in Plant Molecular Biology. And in this case here, the molecules like coniferol alcohol, can be producing lignans in the seed coat, for example, that are in the seeds in the seed coat that go through pinoresinol, but instead of forming plus pinoresinol, they form the opposite antipode to give rise to the minus larisuresinol or plus sequalized larisuresinol, and then through SDG, or in the leaf tissue, they can form plus pinoresinol going through to molecules like yttrium. So let me try and show you this in a different direction here. Sorry, the system is stuck here. It's stuck once, I don't know what its problem is. Yeah, we're also trying to get rid of this bar that's on the computer. If is that blocking out everything? No, you don't see anything, right? Okay, sorry. Okay. So the issue here in flax seed coat is we get coniferol alcohol forming the minus antipode of pinoresinol. It then goes through to this molecule called sequoisolarisuresinol diglucoside, and it then gets decorated, if you will, with hydroxymethyl glutarol moieties. The first one occurs here, the second one is added on there. And then that is further processed with different biochemical pathway intermediates to give rise to mixed pathway oligomers of about 1800 to 2200 molecular weight and size. This thing's frozen. So in the flax seed coat, which is what I'm talking about here just now, it's used, a lot of people in health protecting against breast and prostate cancers. You can take the seeds or the seed coats, you can treat them with base, you can release SDGs, sequoisolarisuresinol diglucoside, treat it with beta-glucosidase, and you can do chiral chromatography and see that it's only the plus antipode that here is generated. We pulled out the corresponding genes, which we call LUDP5 and 6, and these are the ones that generate the corresponding minus antipode rather than the plus antipode that I showed you in the case of podophyllum alexandrum, podophyllum pultatum, and forsythia. So that gives you some idea of the entry point into that, to the opposite antipodes here of pine resinol, and then the types of structures that are generated from there. But we also used multi-imaging mass spectrometry there to localize SDG and the hydroxymethyl glycerol derivatives in the capsules. And here you can see that in the derivative here of SDG, you can see that it's high abundance in the seed coat, but it's essentially absent in the embryo sections. Whereas when we go to the corresponding hydroxymethyl glycerol derivative, you can see that it's high abundance in the seed coat, and again, almost absent in the embryo. And if we take an unknown compound that we just selected out here just for comparison, you can see that unknown is absent in the seed coat, but it's high abundance in the embryo. And then we can look at the overall anatomy of the system. You can see the SDG, the HMG, 6A HMG, SDG, and then we can overlay the two systems that are shown there. And similarly, when we overlay the three molecules, the SDG and the 6-hydroxymethyl glycerol SDG with the unknown, you can get these rather striking pictures. So that took us into one set of class of molecules. Another one that was of greater interest to us were the so-called DP engendered, or what we consider to be DP engendered 804, or in this case 4', 08' lignan formations. A different entry into a different subclass of plant phenols, where you've got this 804 linkage here. And Toshio Mizawa and Kazuki Sato published this in the Plant Cell just in the last few weeks. Toshi used to be in our lab for some time. But while we don't exactly know what the products, what the substrates are for the dirigent protein in vitro in this case, we know from the work they've done with Arabidopsis that this dirigent protein catalyzes a system that enables either the erythro and the thrio forms of these 804 linked systems as shown here. So this brings us to where we've talked about dirigent protein, land plant evolution, and the DP superfamilies. The lignans, pine resinol, and also that 804 or 408 linked lignan I showed you just now are in what we call the Dare family. I'd like to now go on to describe the aromatic diterpenoids, lignans, and then pterocarpon classes of moieties that involve different subfamilies of the dirigent proteins. So Bob Stevanovic down in Texas with Dr. Liu contacted us some years back and said that we think we might have found a dirigent protein, and lo and behold, they had. In this case, it took this achiral molecule, hemigossypol, and in the presence, again, of an auxiliary oxidase, lacase or peroxidase, in presence of this cotton dirigent protein from Thudum, it formed only the plus gossipol, which is much more biologically active than the corresponding minus antipode. So this was the second example of a dirigent protein, but now we've moved from lignans and neolignans into aromatic diterpenoids. But again, radical coupling, but stereoselectively done. Lignification has been a major feature in terms of plants being able to colonize the dry land habitats. And I've mentioned all these different forms that are there. The question has always been in our mind was, how is this system being controlled in planta? So in PNAS a few years ago, Osmani and all, Nico Geldner's lab in particular, were able to show that if you knocked out some dirigent proteins in Arabidopsis root tissue in what they call the Casparian strip, normal lignification no longer occurred and you got patchy or clumpy forms of lignification that indicated that the dirgent proteins, and AT-DIR10 was involved, was the name of the protein, was involved in controlling how the lignin biopolymer was being laid down. So that brought us into another plant phenol class, this case here being the overall lignan moieties. And what they have postulated but have not proven is that there's a scaffold protein, Cas protein, that the dirgent proteins are bound to with peroxidase, lacases, superoxide dismutases, etc., and that this overall complex here is a lignin-forming complex. However, there's been nothing done in vitro to establish what either the substrates are that are being used, what the products are that are being formed, at least on a step-by-step basis, and or whether these protein-protein interactions that are being proposed are anything other than hypothetical at this point. But the interesting thing is with these different different dirgent proteins is they have the same overall fold, but they differ substantially in what we call the beta-1, beta-2 loops. In some cases, they're quite small, 20 to 24 residues, but in the case of lignification, these are much larger. They're over 100 or so residues up there. And again, we don't know what the reasons are for the different loops, but we do know that what appears to be the active site seems to be quite similar in many aspects. So this sort of takes us now in the chronology that we spoke about from 1990 to 2021. And I want to turn my attention now to another class of molecules, the pterocarpins, which many of you will know are phytoalexins or phytoalexin or antifungal compounds. So these are quite interesting. If you look at pterocarpins from licorice, like Glycyrrhiza echinata, it can form molecules like medicarpin. The corresponding enantiomeric or not enantiomeric, different stereochemistries of medicarpin are shown down below here in the corresponding plus form. We can get minus machian in P, Pisum sativum, and we can also get the plus form in Saphoda japonica. And in bean here, you can get physiolin minus form, and you can get pisatin here in P. And I'm going to focus the conversation here today on medicarpin, machian, and pisatin with respect to licorice and also with respect to P and the role of dirgent proteins. So pterocarpins appear to be more or less restricted to legumes. As I said, I'm going to focus on P that produces pisatin plus form minus machian, which have got very different stereochemistries, as I'll point out, and licorice, which accumulates this molecule minus medicarpin. So we pulled out two genes, dirgent protein genes. We call them GEPTS1 from licorice, PSPTS1 from P, and we synthesized the substrates, the potential substrates and products that we call DMI, and you can see the trans and the cis forms. They're either 3R4S or 3S4R, and the corresponding entities in the cis forms, and the products that are potentially generated here in medicarpin. So we purified these recombinant proteins, GPTS1, PSPTS2, and this is shown here, taken to apparent homogeneity, and we then use these for our corresponding assays. So you can see here that when we do a control experiment, incubating with cis DMI or the trans DMI, the cis DMI, I'll need to emphasize, is the natural substrate, and it in the control experiment, you can see both forms of 3S, 4S, 3R, 4R, but in the presence of either the GAPTS1 from licorice or P dirgent protein, the 3R, 4R form is rapidly removed and generates minus medicarpin, whereas if we take the trans DMI, which has the 3s4r and the 3r4s. Again, the 4r moiety is used up by the protein. This is a control. 4r form here is gone, and similarly there. So this is a different mechanism than we saw before, and I'll come back to this again as we go later. In this case, we're not looking at coupling, but the dirigent protein is generating again, or not again, a mono instead of a bis quinomethide intermediate. It's forming a mono equinomethane intermediate, which can then recyclize, or cyclize, should I say, to get entry into this other distinct plant phenol class, the phytoalexin pterocarpin minus metacarpin. We generated the crystal structure of this. This is the licorice form here. This is a trimer of trimers, which is an interesting aspect. We got this down to about 2.6 angstroms. And we also got the corresponding P system, which didn't crystallize as a trimer of trimers. It crystallize as a trimer, but we got that down to 1.5 angstrom resolution. And we can look at the licorice, and we can look at the superposition of a monomer with 6 of these systems and compare it with the corresponding P moiety that's shown there. But the interesting thing is that from docking experiments where we could dock the metacarpin and the DMI, etc., we could come up with 4 potential residues that we thought were potentially involved in enabling this dirigent protein engendered a conversion of DMI into metacarpin. So these are shown here, and as you can see, there are 4 residues: tyrosine 181, tyrosine 103, and 2 asparagine residues at 50 and 83. We mutated these systems into phenylalanine instead of tyrosine, and alanine instead of the asparagine. And then we looked at the kinetic data. Now, I'm not going to go into this in any great detail, other than to say that the licorice and the P system here, in the presence of a corresponding cis DMI, are most active with the cis forms of DMI. But you can see as we do one mutation after another, we don't eliminate the activity of the dirigent protein, but we substantially reduce it with these single mutations. So they certainly indicate that these residues are important in the overall catalytic system that's there. Now, the aspect that I also mentioned to you was that in P, there are 2 pterocarpins. There's a minus form and there's a plus pterocarpin form. The minus form here is Machian, and the plus form here is Pisatin. And you can see the stereochemical differences between these moieties at carbons 3 and 4 here and here, and they differ. There's a methylation here, but there's methylene dialdehyde group formation there, but the other difference is hydroxylation. So the question is, why or how could these 2 systems be somehow generated? So we pulled out, or should I say we didn't pull out, Hans van Etten, the late Hans van Etten in 2014, and Rhodesia Selloy, pulled out a partially purified dirigent protein containing extract from P, and were able to show that this molecule cis-DMDI, which differs from cis-DMI by having this methylene dialdehyde bridge, it would convert this molecule into this achiral compound called DMDIF. And Hans contacted me at that time, just before he died, to say that he thought this was dirigent protein was involved in the formation of this molecule DMDIF. So anyway, we've continued that work with Rhodesia. We've got the recombinant protein that's shown here. We call it PSPTS2. We synthesized again all the various substrates, the trans and the cis DMDI in these different 4 forms, and we're looking at the formation of Machian, plus minus Machian, and also the DMDIF. And this shows the control experiment where we've got the cis and the trans forms here. We can see traces of Machian formation, but in the presence of the cis DMI in particular, we accumulate DMDIF, and we also see that with the corresponding trans form. But when we incubate that same protein with Machian, we convert Machian in that one optical form and not the other one into DMDIF. So what do we think is going on here? We know the substrate here is cis minus DMDI. We know it generates minus Machian, but the minus Machian is then seems to reverse back to forming the monoquinone methide, and that is then after it's ring opened, it then loses, anyway, it taumerizes, loses the elements of water to generate this molecule DMDIF. And we believe that the generation of that moiety there is the substrate that is then metabolized further onward to form a plus pisatin, the other important P, antifungal phytoalexin. But we haven't proven that at this point. So I just would like to finish this up fairly quickly by comparing the divergent protein biochemical mechanisms that we know thus far. So in the first case of either plus affine resinol formation, in the presence of an oxidase and the divergent protein, we form these bisquinone methide. We can get either Ray-Ray or Psi-Psi coupling. We form the bisquinone methide, which then cyclizes to give rise to, with carbon-oxygen bond formation, gives rise to pine resinol. Gossypol is the same process. We get carbon-carbon bond formation here. It can undergo intramolecular cyclization, but it undergoes rearrangement to rearomatize to form gossypol. Similarly, in the 408 lignan class, we can envisage a very similar process. We form free radical forms here at the 8 position, here for the hydroxyl moiety here, and these couple through bisquinone methide, quenched by monoquinone methide, quenched then by hydroxyl through water to introduce the corresponding hydroxyl moiety at this moiety here, but divergent protein engendered. And similarly, as I've talked about the phytoalexins, plus and minus medicarpin, in this case, we're generating the quinone methide. We're not having intermolecular coupling, but we're generating the same sort of intermediate. And then that does the sort of second half of what occurs in pine resinol formation. We get a carbon-oxygen bond formation. And then in the case of the Machian, as I said, to form this DMDIF, this process seems to be reversed, water loss, and then generates the DMDIF, which we believe is converted into plus pisatin. That's later. So the important part here is these divergent proteins are gateway entry points. Lignans is a DIR-A family, aromatic terpenoids, DIR-BD. Similarly, the phytoalexins. We think dimeric stilbenes are in the same class. Lignans are in the DIR-E subfamily. And then the question is, how are the aromatic parts of suberins and cutins, hydrolyzable tannins like elygitannins and proanthocyanidins generated? So these are our future goals, establishing the full range of their physiological functions. And particularly, we're interested in DP-containing supramolecular protein complexes and macromolecular assemblies, whether it's for lignin or for other systems. So we're working with the DOE Joint Genome Institute on a joint genome project. They're synthesizing about 100 to 200 genes for us to express 100 to a couple of hundred proteins in a wide range of plant species that we've chosen from an evolutionary perspective. In the case of looking at a membrane-linked moiety, for example, in lignin formation, we've done some preliminary work in this. We have a paper that's just now out for review, but looking at divergent protein-protein interactions with other systems like oxidases, peroxidases, lactases, and other proteins. But we're very interested in a really two big unknown aspects, and that is, how are proanthocyanidins generated? You can get dimers up to higher oligomer weights, like crofelemer, which is used as an a product to treat diarrhea, antidiarrheal. We don't know yet in any way, shape, or form how these molecules are put together, from the dimer up to, pentamer, up to 30mer, whatever. And then in the case of elygitannins, where you can see the simple form here of telemegrandin, We don't know what controls where coupling occurs at the sites it occurs. And for those that know Elijah Tannin structures well, you know there are a variety of different structural variants that are out there with some rather complicated rearrangements that are almost equivalent to what you see in the alkaloids. So we view these as being two really exciting avenues to explore for other divergent protein gateway entry points into different systems. So just in concluding, the divergent protein subfamilies, the key point I'm trying to make is they're gateway points into distinct land plant phenol metabolic classes. Plant phenol chemistry in many respects has been an understudied system, but I'm hoping that with the divergent protein families that are emerging, this is going to change. The binding pockets in all of the divergent protein structures we know either through homology modeling or through crystallographic aspects are very similar, but they seem to differ, according to what we believe the size of the substrate is. And you can either have carbon-carbon or carbon-oxygen bonding forming processes, but the key aspect here is with quinomethides. We estimate maybe 5 to 7% of DPs have known functions. As I've emphasized already, I don't think we'd have the 390,000 basal plants that went for divergent proteins. They're all trimers, as we can see at this point in time. And we're interested now in looking at higher molecular entities, including lower molecular moieties like simple Elijah tannins and simple proanthocyanidin moieties. These are the folks, Dr. Devin, who's sitting beside me here just now, Jinyang Meng has been involved with Clyde Smith and John Cort and folks from Stanford Crystallographic Center, John Cort from Pacific Northwest and EMSL, and Mike Costa, who does all of our cloning work, Said, who does all our chemical synthesis, Diana does many of the assays that we've done in the past, and Dorlin and Songjun were previous folks that helped in metabolite imaging and also on cloning and expression. So on that, I'll stop, and sorry I ran over a minute or two. Hopefully that gives you some idea as to where these different unusual molecules that we call plant phenols come from. Thank you. Thank you very much.
Datu, please unmute.
Okay, thank you, Professor Norman Lewis, for a very interesting and informative lecture. Now I would like to hand over this session to our MC, Dr. Fatima Salim. Thank you.
Thank you very much, Prof. Dr. Nordin and Prof. Dr. Norman Lewis for your presentation and sharing. And thank you very much, Dr. Lailidin, for assisting the session. Ladies and gentlemen, the time has finally come to our concurrent session. Before that, I would like to invite all the participants to watch Flash Poster Presentation 1 for about 30 minutes, followed by short, approximately 20 minutes coffee break at your own place. And please join us again at 10:50 a.m. sharp for the concurrent sixth session, which is 1A until 1F. The request to join the breakout room can be found on the bottom of your screen, and hope you enjoy throughout our virtual conference. Thank you very much.
Emma?
Assalamualaikum warahmatullahi wabarakatuh. I am Fatih Shazwani. Would like to present my research entitled Comparisons of Boiling and Orbital Shaking Association Method of Vitamin B12 from alveolar Tuka using two-level factory design. So first, this research is mainly focused on two different association methods. We extract vitamin B12 from green edible seaweed alveolar Tuka. Apart from that, we focus on the effect of few independent parameters using two-level factory design. So first we move to the methodology part, which started with the sample collection and drying. And at the same time, we designed a set of experimental design by 2-level factorial. So in this design, 3 different factors were included, and the response was said to be the concentrations of vitamin B12. So after the sample was completely dry, the extractions of vitamin B12 were performed by boiling and orbital shaking following the design created by 2-level factorial design. So upon collecting the extract, they were further purified using Embellex SAD2 packet column for 3 hours, and the purified extract had been analyzed by HPLC afterwards. So the absorbance of 4 vitamin B12 standards, which were methylcobalamin, adenosylcobalamin, cyanocobalamin, and hydroxycobalamin, were measured and compared with the chromatogram obtained for the purified extracts. So lastly, the result was analyzed statistically by ANOVA. So as a result, it was found that a boiled alpha-lactuca contained significantly high cyanocobalamin content, which the retention time was recorded at 1.9 minutes. In contrast, the cyanocobalamin content from orbital shaking extraction method was insignificant. Also, the drying method gave a significant effect on the cyanocobalamin yield, in which oven drying exhibit a positive effect in producing a higher cyanocobalamin yield. So this can be concluded that oven drying may be a viable option for stabilizing alga species in extracting bioactive component. And as for the effect of different factors, it was found that the effect of solvent-solvent ratio was significant at a lower pH, as decreasing the pH will stabilize the vitamin B12. However, the interactive effect between 2 other factors, which portrayed as in figure B and C, was insignificant. It may be due to the solvent to solute ratio has a lower statistical significance in the extraction procedure. Okay, so therefore, in further research, we may suggest to increase the range or to focus on other parameters such as the type of solvent used. So as a conclusion, higher cyanocobalamin yield was successfully extracted from oven dry using boiling extraction method at parameters of 25 to 75% of methanol and water, 3 to 60 gram per ml of solid to solvent ratio, and at pH 3. That's all from me. Thank you.
Good day, everyone. I'm Lokai from Tunku Abdul Rahman University College. Today, I'm going to present on my poster entitled A Study on the Interaction between Apigenin, Camphorol, and 4-hydroxybenzoic acid in xanthine oxidase inhibition. Xanthine oxidase is the main enzyme that takes part in purine metabolism, converting hypoxanthine to xanthine and subsequently to uric acid. High level of uric acid in human body leads to gout disease, which is a painful form of arthritis. Molecular docking is a computer software that is normally used for drug discovery research. Molecular docking predicts the intermolecular forces between ligand and protein complex to determine the inhibitory activity. The more negative the binding affinity, the lower the energy spent to bind to the receptor. The more favorable is the binding mode. The present study is conducted to investigate the interaction between apigenin and camphorol, apigenin and 4-hydroxybenzoic acid, and 4-hydroxybenzoic acid and camphorol in xanthine oxidase inhibition. For molecular docking, the X-ray crystal structure of xanthine oxidase was downloaded from the protein data bank. 2 ligands were docked sequentially into the active site of xanthine oxidase. The output was analyzed and visualized using Autodoc and Discovery Studio respectively. 10 different combinations of ligands were generated and the binding energies were ranked accordingly. The results showed that 4-hydroxybenzoic acid and apigenin were the most stable ligand combination docked to xanthine oxidase. However, when the sequence is reversed, the binding energy increases, which means it becomes less stable. The same phenomena were observed for other ligand pairs, such as the more stable camphorol-apigenin versus the less stable apigenin-camphorol, and the more stable 4-hydroxybenzoic acid camphorol versus the less stable camphorol 4-hydroxybenzoic acid. This indicated that the sequence of docking the ligands into the xanthine oxidase plays an important role in affecting the stability of the multiple ligand protein complexes. The independent ligands dock at non-interfering regional location of the xanthine oxidase active site. Both ligands did not compete with each other for the same cavity, but they block the binding of xanthine substrate, thereby inhibiting xanthine oxidase activity. This suggested additive interaction by the combination of the ligands, in which the combined inhibitory effect of two ligands is equal to the sum of each ligand alone. All right, that's all from the presentation. Thank you.
Assalamualaikum and selamat sejahtera to all the participants. My name is Mazra, a research officer at Forest Research Institute Malaysia. Today's presentation entitled Comparative Biological Activities and Phytochemical Compositions of Different Extraction Process of Melaleuca kerja putih Power. I will discuss some findings on the chemicals and biological properties of selected medicinal freshwater forest species known as Melaleuca kerja putih or gelam. Gelam can be found in low swampy and regularly flooded coastal plains. It has been exploited for the supply of poles and small-scale charcoal industry by the local communities. Although the leaf of gelam have never been used commercially, it has been used traditionally as medicine to treat cholera, diarrhea, muscular pains, scabies, and intestinal worm. Extract and some naturally phytoconstituent found in gelam were reported to have anti-cancer, antibacterial, antioxidant, and also anti-inflammatory properties. Extraction is the main process by which bioactive compound may be obtained from biomass materials. Thus, the objective of this study was to examine the phytochemical compositions of different plant parts of gelam and to compare the biological activities of different extraction process of gelam. For this purpose, the plant material were collected from a selected swarm area in Batu Berendam, Melaka. The samples were then processed, oven-dried and subjected to 3 different extraction processes, which is ethanol, distilled water and supercritical fluids. The resulting extract were then investigated for various biological in vitro models for anti-inflammatory, antioxidants and anti-diabetic properties. This brings us to the result sections. Qualitative determinations of phytochemical showed various plant secondary metabolites in which moderate presence of saponins, flavonoids and condensed tannin in all plant part examined, while the main compounds in the gelam oil were mainly made of melaleucols, beta-cryophyllene, terpinolene, gamma-terpinin and platyphyllols. In another aspect, the bioactivity evaluations of ethanol and aqueous extract of various parts of gelam was conducted using various battery of in vitro assays. Ethanol extracts gave better yield and showed good anti-inflammatory activity in comparison to aqueous extract, while potent radical scavenging activity and anti-diabetic activities were shown by aqueous extract of most plant parts. Water extract also had higher concentration of phenolic compound as compared to ethanol. In contrast, supercritical fluid extractions of the leaf gave moderate anti-inflammatory activity only. As for the conclusions, we found that the extraction solvent had variable effects on the content of secondary metabolite and also bioactivity. Qualitative determinations of phytochemicals showed the presence of flavonoids, saponins and condensed tannins, while the monotropic compositions of the essential oil was mainly made of melaleucols, beta-cryophyllene, terpinolines, gamma-terpinin and platyphyllols. The extract of various plant parts showed variable degree of pharmacological profiles. These findings suggest that both ethanol and aqueous extract are the most potent and could be a promising source of anti-inflammatory, antioxidant and anti-diabetic agent. The results in hand reveal promising activities for gelam for the development of plant-based therapeutic agents or cosmeceuticals. Lastly, I would like to extend my gratitude for all my team members who managed to achieve all the milestones amid the pandemic season and keep things in perspective. The project is financially supported by JPSM under the Technical Research and Development Committee. And with that, I thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nuzawani binti Alias. Today I would like to share with you my works on synthesis, antioxidant, anticholinesterase activities and molecular docking studies of coumaryl 1,3 silnazole derivatives. Inhibition of acetylcholinesterase enzyme is a known procedure to treat Alzheimer's disease. How? The inhibition of acetylcholinesterase enzyme will increase the acetylcholine level in the brain and thus slowing down the progression of Alzheimer's symptom. Why we choose coumarin and sinazole? Because both of these compounds exhibit antioxidant activities, which is important to address one of the contributing factors of Alzheimer's disease, that is oxidative stress. Both of these compounds also possess aromatic ring. Therefore, both compounds can form parallel and strong pi-pi stacks with the aromatic residues at the enzyme active site. In this study, 9 new compounds were synthesized in 4 steps. All of these compounds were tested with antioxidant and anticholinesterase assay. The result shows that all compounds exhibit moderate to weak antioxidant activities, with compared to the standards, with 6E and 6F exhibit stronger antioxidant activities compared to all other derivatives. All compounds also exhibit moderate to weak anticholinesterase activities compared to standard physostigmine, with 5C and 6B exhibit stronger anticholinesterase activities in their series. So both of these compounds were further analyzed for their binding interaction with the enzyme using molecular docking. So this figure 3 shows the binding interaction of 5C and N-acetylcholinesterase enzyme active site. As shown in table 1, both of 5C and 6B exhibit pi-pi stacks with the aromatic residues at the active sites of the enzyme, similar with donepezil. Therefore, this shows that although these compounds exhibit moderate to weak antioxidant and anticholinesterase activities, they form pi-pi stacking interaction with aromatic residues at the active site of the enzyme, which is responsible in inhibiting the enzymes. The synthesized compounds contain skeletal structures that can interact with the enzyme active sites and therefore inhibit them. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. A very good morning to all of you. Today I would like to present my study about the identification of subgingival plaque bacteria in periodontitis patient with diabetes mellitus using 16S RNA method. For objective, the study is to identify the bacterial type in a subgingival plaque of periodontitis patient with diabetes mellitus. We also have several of specific objective, which is first, we would like to determine colony forming unit of bacteria in subgingival plaque of periodontitis patient with diabetes mellitus. Second, to isolate the DNA extraction of bacteria in subgingival plaque in periodontitis patient with diabetes mellitus. And third is to determine bacteria species of subgingival plaque in periodontitis patient with diabetes mellitus. We have also have several methodology to achieve all the objective. First, we collect the subgingival plaque sample in periodontology and community oral health department, Faculty Dentistry USIM. Second, we do a serial dilution before we grow to the blood agar, and then we observe CFU per mil. And then we observe also the morphology using a microscopic analysis. And fourth, we do a molecular analysis, which is we screen the DNA isolation, and then we also sequence the DNA, and then after that we analyze the sequencing using the BLAST-N. For the result, we only got 2 samples of the plaque, and then we do morphology identification using a blood agar, and we can see morphology of the cultures, such as the culture is small and they have translucent, and the surface is smooth, and the color is white or gray. After that, we do a gram staining, and then we can see the mixture of the culture, and also we can get a pure culture, and from the pure culture we isolate the DNA using the heat shock method, and then after that we run using the PCR electrophoresis, and then after that we send to the sequencing analysis. After that, from the sequencing analysis, we can see several of number in the in the analysis, and we got a gram-positive bacteria were found more compared to gram-negative bacteria, and then we also found periodontal pathogen such as Agrobacter actinomycomitans, and also we also found a Porphyromonas gingivalis. And the conclusion is there is a presence of variation of bacterial species in subgingival plaque of a periodontist patient with diabetes mellitus, whereby gram-positive enrobe were found more compared to gram-negative enrobe. We also found non-oral bacteria in periodontist patient, which is we found Enterobacteriaceae species. Thank you for the presentation. Assalamualaikum.
Today, I would like to present on my poster, Medicinal Hemp Research, Trends, Gaps and Challenges. This poster is part of a larger scoping review conducted on medicinal hemp by my team. Medicinal cannabis is a controversial topic which draws the attention of many. Based on the tetrahydrocannabidiol THC content, the cannabis plant can be broadly categorized to either marijuana, which is famous for recreational use, and hemp, with limited THC concentration of less than 0.3%. THC is the main psychoactive component of cannabis and is responsible for many unwanted effects. Hemp fibers and seed were mostly for industrial use. In the past few years, there has been increased interest in the health benefits of hemp, with many beneficial claims on the internet, such as a sleeping aid. As medicinal hemp research is a new but rapidly growing area, this review was conducted to identify current research trends, gaps, and challenges on medicinal hemp research in the past decade. We searched 4 major databases using pre-specified keywords combination. From the 11,305 records initially identified, we narrowed it down to a final 65 included articles. All these articles reported on hemp-specific research. Figure 1 shows the rapidly rising trend of published medicinal hemp research. There is an evidence search from 2018 onwards, representing recent growth of interest. In 2019, the number of clinical papers more than tripled the papers in 2018. The pie chart in Figure 2 depicts the country distribution of published medicinal hemp research. Here, we can see China and Italy as the forerunners. Figure 3 illustrates the most commonly investigated indications and pharmacological activities of hemp in clinical research. The studies on the top levels of the diagram are of higher level evidence, while the level of evidence reduces as we go down the diagram. Interestingly, hemp is the most commonly investigated to relieve constipation. In conclusion, there is a growing number of studies conducted on hemp and hemp-derived compounds. Most hemp-specific evidence is preliminary with insufficient high-quality evidence for any specific therapeutic indications. There is limited quantitative data and uniformity in the contents of hemp extracts and formulations investigated. As hemp is highly varied in both cannabinoid and non-cannabinoid contents, there is a need for proper authentication and focused selection on hemp formulation as test items in biomedical research. Improved methodological design is an important factor in strengthening and focusing future hemp research.
Imagine when you are walking on a roadside, you find a beautiful plant. You impressed, but then you just continue your journey. Tomorrow, when you walk on the other roadside, you find a beautiful plant that looks similar with yesterday. But wait, are you sure they are same species? This question led me to this experiment. From the day I found this plant, I believe that I must get the anatomical and micromorphological data for this plant. Since anatomical and micromorphological research in plants is still new in Malaysia, I believe that I can get interesting data from this research. In the future, if pharmacologists want to create new medicine, but they have only leaf on their hand, how can they be sure the leaf on their hand is Youngia japonica species? Thus, the result from this experiment can be used by them to identify the leaf that they have at that time is Youngia japonica species. After they get the fit characteristic of the leaf, thus they will not mislead the plant as another plant that looks similar on the morphology. Based on these four methods, which are cross-section, peeling, clearing, and scanning electron microscopy, or known as SEM, can be used to identify a plant. As we can see here, the shape of the petiole and the shape of the vascular bundle can be identified as Youngia japonica species. The shape of vascular bundle is open system that arranged in arc shape. The shape of the leaf margin look downward with blunt end. Another result for Youngia japonica, the type of stomata is anomocytic, means the stomata have no subsidiary cell around the stomata. Also, the incomplete venation and the waxes that found on the leaf shows the result of Youngia japonica. And last but not least, the trichome that obviously known present in Asteraceae family. That's all from me. Thank you.
Kita indah di mata dunia, hidup harmoni berbilang bangsa, wajah riang manis senyuman, sejahtera sealun senada sejiwa. Pantai pasir putih, lautan biru, desiran ombak dihembus bayu, hutan hijau malam berseri, bunga-bungaan harum mekar menawan hati. Malaysia indah, nadi hati jiwa kita semua. It's a truly Asia, Malaysia, warna-warni kehidupan bahagia. Malaysia, melawat Malaysia, tentang seni budaya dan bangsa. Visit truly Asia, Malaysia, segala bermula di sini, di Malaysia. Adik kakak buat patah bagi Malaysia.
Everyone, I dah disconnect